Good morning, students. Today, let me continue with that organic evolution chapter because in that previous class we have discussed about Lamarckism and Darwinism. And in that Darwinism, there were some demerits or defects were existing, and those things, some of which we already discussed in the previous class itself. And today, let me just continue with the demerits of Darwinism first. So, Charles Darwin, that is, natural selection theory, is favored by many biologists. Still, there are criticism or there are some lacuna in it, and they are: Darwin did not explain the actual process of inheritance of characters as it was explained by Mendel. Darwin proposed the theory of pangenesis to explain this phenomenon, and he said that every cell or organ produces minute hereditary particles called pangenes or gametes, and these were carried through the blood and deposited in the gametes, and then it will be transferred to the next generation. So this is how the inheritance pattern was explained by Mendel, but there is no proof for the existence of such pangenes, so this theory was not accepted. So this is one major big lacuna that is present in the Darwinism. And he did not explain the origin of new characters. That means Darwin never explained how new characters are going to be appeared, but he just said that characters will appear, new characters will appear, but there is no substantial evidence for that. And according to natural selection theory, only useful organs are favored by natural selection, and the existence of vestigial organs in organisms could not be explained. You know what are vestigial organs are? And though these vestigial organs do not have any function, but still they are present in different animals, and there is no explanation given by Darwin with regard to these vestigial organs. So though these vestigial organs are present, but there is no substantial explanation given by Darwin regarding that. So this is another drawback or demerit of Darwinism. And Darwin theory fails to explain the source of variations. So in the previous class we have discussed that in Darwinism he just said that variations will appear, but he never explained how these variations are produced and what could be the cause for these variations. And the later, the post-Darwinians, they have convincingly proved that how these variations are produced. So this is another drawback or the demerit of Darwinism. And there is one more such demerit of Darwinism, that is, Darwin never distinguished between heritable and non-heritable variations. So what Darwin said, he said only the appearance of variations, and he never made the distinction between heritable and non-heritable variations. That is, whether all variations will be heritable or not, that explanation was not given by Darwin. So that could be one of the demerits of Darwinism. And the post-Darwinians, or who are also referred to as NeoDarwinians, they have proved, they have given the explanation for all these demerits in this new concept, what is called as synthetic theory of evolution, or it is also called the genetical basis of organic evolution. The reason is, after 1900, intense work has been done in the field of genetics after the rediscovery of Mendelism, because of which now today we have the good idea about how variations are produced, that is by the advancement in the field of genetics. And as new ideas were synthesized after Darwin, this theory is also called synthetic theory of evolution. Okay, and this Neo-Darwinism, it is called so because neo means new, as you know. It is a new version of the existing Darwinism. That means though there are demerits in the Darwinism, it was not completely rejected, They have retained some of the principles explained by Darwin, but they have given a new shape to the Darwinism by means of adding some new concepts or the new principles to it. Hence, the phenomenon or the concept is also called NeoDarwinism. So, this is having different names. See, as I told you already, it is also referred to as genetical theory of natural selection or Neo-Darwinism, and it is a modern concept of organic evolution which has been developed by renowned scientists like Chivalright, Fisher, Dobzhansky, Muller, Huxley, J.B.S. Haldane, Stebbins, Ayala, etc. So it was not proposed by one scientist, but the ideas generated by different scientists have been combined together. Hence the concept is called Neo-Darwinism or synthetic phase of Darwinism or synthetic theory of natural selection. And it is a new approach to the organic evolution in the light of new branches of biology like genetics, cell biology, molecular biology, etc. And according to this theory, sexual reproduction plays an important role in evolution. As you know that during sexual reproduction there could be the appearance of many number of variations when compared to the asexual reproduction. That means during asexual reproduction the chances of production of variations are very less, but they are very more in sexual reproduction. Hence, sexual reproduction plays a very important role. And so far as evolution is concerned, a population consists of genes of all the individuals in the population, and this sum or the total number of genes present in such a population is referred to as a gene pool. So what is gene pool? It is the sum total of all the genes present in a given population at a given time, which reproduces mainly by sexual method of reproduction. So, gene pool is defined as the sum total of genes occurring in a population of sexually reproducing organisms. Hence, the gene pool and additions or deletions of genes from the gene pool are the focal points of synthetic theory of evolution. Some biologists have refined evolution in genetical terms as a change in the gene frequency, and gene frequency is merely the frequency of an allele or a gene in a gene pool, and changes in the gene frequency results in variations, and variations leads to the development of a new species. We may list 5 such evolutionary forces that change the gene frequencies. They are mutations, gene flow, genetic drift, isolation, and natural selection. Coming to mutations. You know what are mutations and who discovered it? Mutations were first discovered by a scientist by name mutation theory. You know who has discovered these mutations for the first time and who proposed it for the first time? That concept of mutation theory. And mutation is a sudden heritable change that occurs in the genetic makeup of an organism. And Dobzhansky stated that mutation is a mistake or misprint in cell division, and the mutation produces a variation which is the cause of organic evolution. And there are various types of mutations which are as follows. Number 1, dominant mutation. When mutation produces a dominant gene, it is called dominant mutation. And dominant mutation expresses the character immediately. Whereas recessive mutation is that when mutation produces a recessive gene, it is called recessive mutation. And most of natural mutations are recessive in nature, and the recessive mutation do not express the character immediately, unlike it is present in dominant mutations. But it is expressed only when the mutant gene occurs in homozygous condition or in double dose condition, as we have already discussed about this in hemophilia, and in case of that color blindness disease. And gene mutation. The sudden heritable change of a gene from one form to another form is called gene or point mutation, and it is called point mutation because it affects only a particular locus in the chromosome, and the gene mutation is classified into different types which are named as follows. So though these mutations were first discovered by Hugo de Vries, a Dutch botanist, but it was implemented successfully in this field of organic evolution by the NeoDarwinians. Coming to the first type of mutation, that is deletion. So you know what is deletion is? It is the loss of one or more nucleotide from a gene is called deletion. And a nucleotide is a fundamental unit of DNA and RNA and are composed of a nitrogen base which is attached to a sugar phosphate unit. Thus, nucleotides are the building blocks of nucleic acids. Coming to insertion, the next type of mutation. As the name itself suggests, it is the addition of one or more nucleotides to a gene. So, if one or more nucleotides are added to a gene, it is called insertion. Then about inversion. In this case, as the name itself suggests, a segment of DNA is removed and reinserted in a reverse direction. So it is just the inverted sequence of some nucleotides of a DNA molecule. Then about substitution. In this case, one nucleotide is replaced by another nucleotide, and it is of two types, that is transversion. It is the substitution mutation where purine, that is either adenine or guanine, is replaced by pyrimidine, that is cytosine or uracil, and thymine, or vice versa. Then about transition, the next type of substitution mutation. It is a substitution mutation where purine is replaced another purine, or pyrimidine is replaced by another pyrimidine. So such type of mutations are called transitions. And mutations are considered to be the raw materials for evolution, one of the major factors. And they are the sources of all genetic variation in a population or a gene pool. The mutation process constantly generates new hereditary variants, and these variants accumulate in the population. The mutation may be useful or useless, or harmful or neutral to the population. The population will be benefited by the production of useful mutations. So, mutations are the sources of evolutionary forces that change the gene frequencies, which in turn the cause of evolution. So one of the major factors for origin of variations is mutations. Coming to gene flow. Transfer of genes between populations which differ genetically from one another but can interbreed is called gene flow. Or it is the flow of genes from one gene pool to another gene pool. Thus, it becomes an important and quick way of causing changes in gene frequency. Gene pool is defined as the sum total of genes occurring in a population of sexually reproducing organisms, which we have already discussed earlier. So, the gene flow occurs when individuals migrate among the populations and they contribute their genes to another gene pool. The gene flow increases the amount of variability within the population. For example, the migration of Africans and Indians to USA has resulted in the change of gene frequencies of white human population. And a change in the gene frequency in a population will thus depend on the rate of gene flow from another population. So this is about the gene flow, and now about genetic drift. It is also called as fluctuation or random selection. See, This natural selection usually it operates on large sized population, where it always favors the beneficial characters, and it is not going to it is not going to favor the non-beneficial characters. Whereas, unlike in natural selection, in random selection, or what is called as genetic drift, it operates in a small sized population, and it selects the individual at random, whether it is useful or useless, or whether it is harmful or harmless. So it selects the individuals at random, hence it is also called random selection. And when a population is small, there is a greater possibility of chance causing changes in the gene frequencies. Through genetic drift, some alleles become established and others are lost. And this happens irrespective of its survival value. Supposing there is a population of blue parrots and green parrots, and the allele controlling blue feather color is lost as a result of genetic drift, all the parents of the next generation will be green only. And this change has not arisen because green feather color is more advantageous, but because chance events led to a change in a genotype. And Civil Wright was the first person to recognize that genetic drift could change gene frequencies of a population. Hence, genetic drift is also called Civil Wright effect. So, what is Civil Wright effect is? Civil Wright effect is nothing but the genetic drift only that operates in a small population, which always change the gene frequency of a population, thereby leads to variations, and ultimately it promotes to the evolution. Coming to bottleneck effect or phenomenon. This term was coined by Stebbins, and hence it is also called Stebbins effect. See, the environmental fluctuations like shortage of food or outbreak of disease may bring about a marked decrease in the number of individuals, as it is happening with reference to this COVID-19 disease. Then the population goes through a bottleneck effect. That means in the peak level of this infection or the disease, more number of individuals will die, and very next time the number of such individuals will increase. So it is like a bottleneck phenomenon, hence the concept is called bottleneck effect. And then the population goes through a bottleneck effect, and the yearly cyclic phenomenon of population is called bottleneck effect. Let me quote an analogy here that these mango trees, they will produce more yield in 1 year, but with the very next year the yield is quite less, or comparatively very less. So this shift is seen in terms of the production of mangoes by mango trees. We can also call it as the bottleneck phenomenon. And Stebbins effect is the transformation of larger population into smaller population and vice versa. So if you upright the bottle, the base will naturally contain more content there, whereas in the neck region it will be very less. Hence the concept is called bottleneck phenomenon. Examples: lemmings are small rodents of Scandinavian mountains, and these rodents show bottleneck phenomenon. In every 4 years, the population of these lemmings reaches an unbearable maximum level, and at this maximum level, the lemmings migrate to the coast, plunge into the sea, and committed suicide. And the population is re-established by the few which remains in the mountains once they return back to their native place. So this effect is one of the best example for this bottleneck phenomenon. Coming to isolation. Isolation is the separation of individuals of a species by some barrier which prevents interbreeding. So if interbreeding is prevented, naturally there is no scope for variation, but isolation brings a significant change here. So separation of a single population into subunits is called isolation, and when interbreeding is prevented, gene flow between populations is also prevented. And the isolated groups do not interbreed, and each group may develop a set of new characters which leads to the development of new species. The term isolating mechanism was first coined by Dobzhansky in 1932, and according to him, without isolation, organic evolution is not possible. And the mechanism by which interbreeding is prevented is called isolating mechanism. The isolating mechanisms are of two types, namely geographical isolation and reproductive isolation. Coming to the first type, geographical isolation, it is the separation of two populations by geographical barriers such as landmass, oceans, mountains, deserts, and thick forest is called geographical isolation. Thus, there is no possibility of interbreeding between isolated groups, and the isolated groups in course of time forms two different species, and the Indian giant squirrel Ratuffa indica lives in forests. It is yellowish in color in the deciduous forests of Gujarat, light brown in the deciduous forests of Maharashtra, and deep brown in the evergreen forests of Mysore. Here, forests act as barriers, and this leads to the development of subspecies with different colors within a species. So this is one of the best examples for geographical isolation. Coming to reproductive isolation. This is the separation of populations based on structure, function, and behavior that prevents the interbreeding between populations. The following are the various types of reproductive isolating mechanisms, which can be broadly categorized into or classified into two types, pre-zygotic and post-zygotic, but here these types are given as follows. Coming to the first type, pathological isolation. In some species, differences in sexual behavior prevents interbreeding between individuals. See, as the name itself suggests, ethology, that is the study of behavior, so that is the sexual behavior is different, and that separates the interbreeding between two different populations. And this is called ethological isolation. For example, Drosophila melanogaster and Drosophila simulans have different patterns of courtship movements. So, interbreeding is not possible between them. Next one is seasonal isolation. Here, interbreeding is prevented because of differences in the breeding seasons. For example, Amblyostoma tigrinum and Amblyostoma maculatum, which lays in the same area but do not interbreed, as Amblyostoma tigrinum completes breeding before March, while the Amblyostoma maculatum breeds after March. So because of this difference in the seasons, these two species of Amblyostoma cannot interbreed. Next is habitat isolation. Here isolation is prevented because of different habitats. Water snake, for example, that is Natrix hypendon, lives in fresh water and also in salt water, and they do not mate with each other because of habitat differences. Then coming to natural selection. Natural selection is an evolutionary theory proposed by Charles Darwin, which we already discussed. It explains the mechanism of evolution and the origin of new species. The natural selection operates by selecting favorable organisms and eliminates unfavorable organisms. And the best experimental support for this operation of natural selection was provided by H.B.D. Kittlewell in a study of industrial melanism. Kittlewell studied natural selection in the peppered moths, that is Biston betellaria. Before industrial revolution in England, about 130 years ago, all the peppered moths lived in Great Britain were white colored. And the white color was due to the absence of melanin pigments. And they lived in white background of the lichens on the bark of the trees, and the white color of the moths is matched with the background of the white lichens, so birds could not be picked and eat these white moths. And the white body color was favorable variation at that time, and this variation was selected by the nature, and the non-melanic forms or white colored moths survived before 1845. However, in 1845, one black moth or melanic form appeared, and this was due to a single mutation. And this black moth was called Biston carbomaria. And in 1848, the black variety forms 1% of the total population from one number, but in 1898, the black variety formed 99% of the population and only 1% of Biston betullaria. And after 1848, more and more industries were developed in Britain, which we call it as Industrial Revolution, and the smoke and soot arising from the industries were deposited in the surrounding area, including the bark of the tree. So the bark of the trees become dark, and now under this background the white or non-melanic moths were easily picked and captured by birds. And in this situation, white body is an unfavorable variation, and this variation is rejected by the natural selection. On the other hand, the black moth now had favorable variation and is selected by the natural selection. This Kittlewell was a very close observer of this phenomenon, and this could be one of the best examples to illustrate the mode of operation made by the natural selection. And Kittlewell took melanic and non-melanic moths and distributed them in highly industrialized city Manchester and non-industrialized city, that is Dorsal. And after some months, he captures them and found that more than 90% were melanic forms from the Manchester and more than 90% were non-melanic forms from the Dorsal. So this is because of the industrial smoke or the soot released by the industries. Thus, industrial melanism clearly explains the operation of natural selection. And according to Johansson, natural selection is effective in genetically mixed population but ineffective in genetically uniform population. And mutation causes variations which may be favorable or unfavorable or neutral. And the animals provided with favorable variations are well adapted in the given environment, and the environment also favors the individuals which are provided with favorable variations. So, natural selection favors the genotype which has maximum adaptive value. So, this is the adaptive value or the Darwinian fitness of the genotype. And thus, natural selection fixes up favorable variations and eliminates unfavorable variations. So this could be one of the best example for natural selection. And another example of natural selection is insecticide resistance in mosquitoes, particularly with reference to DDT. Mosquitoes are the vectors of diseases such as malaria and filaria. In the 1950s, they were controlled by the extensive use of pesticide, that is a chlorinated hydrocarbon, namely DDT. And its use was so effective that India had cut the incidence of malaria to a negligible level. However, in 1970s, malaria came back and it was found that DDT was ineffective against mosquitoes. How can this be explained? In the original mosquito population, there were already some mutant individuals which were resistant to DDT. However, their number was small. And when DDT came into use, they had a distinct advantage over the sensitive mosquitoes. Hence, they were able to survive and reproduce while the sensitive ones died in large numbers before they reproduced. So, this is what the difference observed with reference to their reproductive fitness, which is called reproductive isolation, or what is called as differential reproduction. That is, the sensitive forms could not reproduce, whereas the resistant varieties could reproduce. Hence, it is called differential reproduction. And as a result, over a period of time, the resistant population increased, and this made the DDT ineffective. Hence, a change in the environment, that is the introduction of DDT, was able to bring about a change in the frequency of alleles by differential reproduction. And gradually the genotype structure of the population shifts towards more resistant phenotypes. And insecticide resistance is another good example for natural selection or directional selection. Now, coming to types of natural selection. The three types of natural selection are stabilizing selection and directional selection, and the next one is the disruptive selection. Let us go through the stabilizing selection first. It is a type of natural selection that favors the average individuals in a population. For example, for a plant, the plants that are very tall are exposed to more wind and are at a risk of being blown over, while the plants that are very short fail to get enough sunlight. Therefore, the plants that are a middle height between the two get both enough sunlight and protection from the wind, and they have stabilized in a population, and this is what called stabilizing selection. Whereas the second type is directional selection, and in this type of natural selection occurs when selective pressures are working in favor of one extreme of a trait. For example, giraffes with longest necks are able to reach more leaves of a tree, and selective pressures will work in the advantage of the longer neck giraffes, and therefore the distribution of the trait within the population will shift towards the longer neck trait. That is, it is a directional selection which allows the improvement or the increase in the number of such giraffes in one direction, that is having long necks. Coming to the third type, disruptive selection or diversifying selection, and this type of natural selection occurs when pressures are working in favor of the two extremes and against the intermediate trait. For example, an area that has black, white and grey bunnies contains both black and white rocks, and both the traits for white and black will be favored by natural selection, since they both prove useful for camouflage. And the intermediate trait, grey, does not prove as useful, and therefore selective pressures act against the trait. So this is with reference to disruptive selection, which is also called diversifying selection. And this slide will show you how that natural selection favors the favorable variants. As you see here in this picture, before industrial revolution, these non-melanic moths, that is betel area number is more, and there were only few number of Biston carbomeria, whereas after industrial revolution you see there are more number of melanic forms than non-melanic forms. So this could be one of the best example for the natural selection, which I have already explained to you, and this is for your reference. And this is again a good example for natural selection, that is about sickle cell anemia. The sickle cell anemia are the anemic people, that is in tropical areas of Africa. They were resistant to this disease, what is called as malaria. You know why? The sickle cells will not allow this pathogen, the protozoan pathogen, to multiply, and it will actually kill that particular parasite in the sickle cell shaped RBCs itself. So they are said to be resistant against malaria. So having one disease, that is sickle cell anemia disease, will favor the person to avoid the malaria. So it is a very unusual thing that is observed, that one person having the sickle cell anemia will not get the malaria very frequently in such populations. So this is another good example for natural selection. So natural selection here, it favors the person having sickle cell anemic condition than the normal shaped RBCs. So this is again a photograph, the graphical representation of these different types of natural selection, which I have already explained you, like stabilizing or balancing selection, directional selection, and disruptive selection. And these graphs are for your additional information. So with this, I have completed this topic, that is Neo-Darwinism. Thank you.